Hi, Chapter 12 deals with modern applications of using microbial genetics and some of the techniques that are used for that. Recombinant DNA is something that you hear a lot about. We're using technology to help manipulate the DNA sequence, so you end up with new combinations of the DNA. So the recombinant DNA is one that has new combinations. Basically, it contains some genes or information that it did not originally have in there. Transgenic means that it contains DNA from a different species. An example of this is the bacteria E. coli, where we have put the human growth hormone gene into it. That was obviously not originally there. And so we have added that new combination of genes to it to produce a certain product that we're wanting. Molecular cloning. We're going to look at some of these techniques where it's going to involve restriction enzymes. Restriction enzymes are enzymes that are going to cut the DNA in very specific places. It recognizes a certain base sequence. Usually, most of them recognize about 6 bases. And it has to be in that particular order of those bases, and then it will do a cut. It will either do what we call sticky ends or blunt ends. We use this quite a bit. There are hundreds of restriction enzymes that if you can, it's a way of cutting the DNA so you can insert something into it. We work a lot with plasmids. We also work a lot with ligation, which is binding the fragments back together of the DNA. So with restriction enzymes, excuse me, the way it works is up here you have a strand of DNA, and for a sticky end type of cutting, it would recognize this base sequence and it would cut right here. And then it's going to cut right here as well. So you're cutting right here and right here where it's indicated. So you end up with these ends, but we call this end, because this has exposed ends right here, we call them sticky ends. If you have a preference and a choice, this is what most people are going to prefer. It just makes it easier to stick your new DNA piece in. So what you're going to do when you mix it, you would cut the recipient DNA and the donor DNA using the same restriction enzyme. And so then the piece you want to stick in here, it's going to be complementary to here, and it'll attach right there a little bit easier. Blunt ends, you're just going to do a straight cut across, and that's it. That's what you end up with. With molecular cloning, what we're talking, you're going to usually take plasmid DNA, cut that, and then you're going to cut the DNA that has the desired gene in it. Like I said, you use the same restriction enzyme. Once you have cut them, you usually put them in a flask together or a beaker together, and you mix them. You allow them to bind together. Then you're going to add DNA ligase. Remember, you saw this with DNA replication. Excuse me, this is where it kind of is that DNA superglue, if you will. It will connect the fragments together. So now you have a plasmid with the extra piece of DNA in it. You're going to now take that plasmid, add it to the host cell. And you need to have some way of monitoring or observing to see, was it successful in getting it in the host cell? Do you get the product that you want? And so this is just saying, here, in this case, it's a human cell. Here's your DNA. You removed, in this case, it's removing the insulin gene. So that would be the gene that triggers the production of insulin. The blue is your plasmid. You have taken a restriction enzyme and you have cut the plasmid and the DNA using the same restriction enzyme, so that the ends, the exposed ends here, are going to be complementary. And then you mix these two components together in like I say, in a beaker and a flask together. You allow them to recombine. You add the DNA ligase so that it connects and seals the connections here between them. And then you take this plasmid and you put it into your bacteria. So this is now considered a transgenic bacterium, like we said earlier, such as E. coli, because you now have a piece of DNA in here, this little red piece on the plasmid, that doesn't normally exist in this organism. It's from a totally different organism. So you grow it, and now what happens is all the offspring of this, it's producing insulin. So you're able to collect the insulin, filter it away from the bacteria, and you now have human insulin that you can give to diabetics, which is certainly going to be a lot better for the diabetics. Before they could do this, they used to use pork insulin, and sometimes you had reactions to that. So this is a process, once again, of showing you have your foreign DNA here, you have a plasmid, you're going to cut using restriction enzymes, you stick. Now in this case, you're looking at several different genes potentially, putting those each into bacteria. Now you've got to figure out, when you grow the bacteria on a plate, which one has which gene. And that's why you always have to think ahead in your experiments. So how are you going to identify which bacteria has which gene? Genomic library is just referring to generation of what we call molecular cloning library, where you have a complete copy of the organism's genome contained in plasmids. And then you can take the fragments. You use that to determine the sequence of the DNA and what's each gene's purpose, what's the function of it. This does allow for you to produce large quantities of a particular DNA fragment. So you've taken the entire DNA, broken it up. Oh, okay, I need fragment D. And so in this case, it's showing you took DNA from the organism, excuse me, you cut it into fragments, put those fragments in plasmids, put the plasmids in bacteria. So now over here you see you have this whole library. Each one of these is going to produce new ones with the same fragment, same plasmid in it, with that fragment of DNA. So you can figure out, okay, if we use the colors here, oh, the red fragment is a gene for such and such, but the orange one is for something else, and the green one, that's why you can figure out what does each gene function for. Oftentimes when you're doing things, you're going to read about complementary DNA or cDNA. And what this complementary DNA is, the way we make it is we use mRNA as a template to make it. Now, in transcription, you use DNA as a template to make your RNA. This is going in the reverse direction. There's nothing that says you can't do that, and indeed you can. You use an enzyme called reverse transcriptase, so it's working the reverse way. So it's going to use the RNA as a template to make the cDNA. And how is the cDNA different from the original DNA? Remember that the eukaryotic DNA has those introns, those little sections of extra DNA that it has to cut out during, it goes through transcription, but at the end it has those modifications where it has to cut those out, and then it splices together the exons. Prokaryotic cells do not have introns. And so if you want to take DNA from a eukaryotic organism, such as humans, say like the human insulin, and you want to put that into a prokaryotic cell, such as E. coli, to make a particular product, there's going to be a problem. If you put the DNA straight into a prokaryotic cell, because eukaryotic cells have introns, that DNA is going to have the introns in them. Prokaryotic cells don't have that. They don't know, oh, wait a minute, I'm supposed to go ahead and carry out transcription, but afterwards I need to cut some of this extra DNA, these little introns, I need to cut them out. They don't know to do that. They've never had to do it before. It'd be like, telling a kid to do something, well, what are you talking about? So they don't know to do that. And so it's not going to express the gene. You're not going to get the product. Say in this case, you would never get human insulin made. And actually, this is how they found that the introns existed. They tried putting the gene for human insulin into E. coli, and it wasn't put in there. They had their markers to see, yes, we put it in there. Why is it not producing? And then finally someone suggested they kind of work backwards. And that's when they found out, oh, guess what? I guess eukaryotes have these little extra pieces called introns. Prokaryotic cells don't. So what they realized, the way around that was, you take the original eukaryotic DNA that contains the gene of interest. You let transcription occur so that you get the mRNA, the mature mRNA, after all the extra splicing has been done. Then you take that mature mRNA and you use that as a template and work backwards by using reverse transcriptase to generate your complementary DNA, your cDNA. That DNA does not have the introns in it. Now you can put the cDNA into a prokaryotic cell and bingo, you're going to get production of your product. You will get expression of it. So it's just something to remember if you're going to try to insert a eukaryotic DNA into the prokaryotic cell. It has to be the cDNA. It's not going to work otherwise. And so, once again, just showing you use that reverse transcriptase, you get the cDNA, stick that in the bacteria, and now bingo, you get your product. So how do you introduce DNA into prokaryotic cells? We've touched on this briefly before. There's three main ways. There's transformation, transduction, and conjugation. Transformation is when it just uptakes naked DNA that's present in the environment. Transduction is when you're going to use a vector, and that vector is going to be a virus that will introduce your DNA into the prokaryotic cell. Conjugation is only going to occur with gram-negative bacteria because it's going to use pili, and pili are only produced in gram-negatives. And the two cells have to be compatible. So you have to have a lot of factors in place in order for the conjugation to work. The pili will connect from one cell to the other. It's like a hollow tube, and the DNA will go from one cell to the other. So how do you introduce DNA into eukaryotic cells? It's by a method called transfection. There's several different options here. Electroporation, gene guns, microinjection. You can still use viruses. You can use shuttle vectors. And so electroporation is simply where you've got your cell. You have the purple's your recombinant DNA. You're trying to introduce some of these genes into the cell. You add an electrical current. That's going to create these pores that will open up. It's kind of like these, it's kind of like, oh, let's open the windows and let some fresh air in. So it's going to just very temporarily, as long as you have the electrical current going, it will open these doors that will allow the DNA on the outside to flow in. Things naturally want to go from high concentration to low concentration. It's going to flow in. As soon as you turn off that electrical impulse, it's like shutting the windows. These pores are going to close, but guess what? Now you've got some of those genes inside the cell. So that's one way. Another way is to use a gene gun. This is where you have projectile, you coat like these little, they actually look like small little bullets with your DNA on it, and you literally fire it into, in this case it's showing with a plant cell. Another way is microinjection. This is fairly popular, where you have microinjection needle. It's a very fine needle. You are working through a microscope in order to see basically what you're doing. You have the needle and you will actually inject into the cell, and you want to inject into the nucleus, and you're injecting the DNA, donor DNA, into here. So like I said, there's several different methods. Here's a shuttle vector. You're using oftentimes plasmid that's commonly found in Agrobacterium tumefaciens. This is used in agriculture a lot. Agrobacterium tumefaciens is a bacteria commonly found in the soil, and the TI plasmid is a plasmid that's typically found in it. And so you're using that. You stick your gene of choice that you're wanting to add, say, to a plant. You stick that on the plasmid. You put the plasmid back in the agrobacterium, and then you use that to infect the plant. How do you analyze the DNA? How are you trying to analyze? Did you get the piece of DNA into the recipient cell that you wanted? How do you know it's there? There's several different ways to measure that as well. You can use nucleic acid probes, agarose gel electrophoresis, restriction fraction length polymorphism, which is RFLP. You can do a southern blot. Just so you know, it's kind of strange, just sort of FYI, but a southern blot, you are looking at fragments of DNA. That was the first type of blot that was done, and actually the individual who developed it, his last name was Southern. So then somebody then decided, well, what if we do a similar thing but with RNA? Well, we can't give it the same name because DNA and RNA are not the same thing. So a southern blot is with DNA. A northern blot is with RNA. And I don't know whose idea, I guess they just decided in a comical fashion to, oh, well, the first one's southern, then we go with northern. There is also a western blot, which is with protein. So southern blot is DNA, northern blot is RNA, western blot is protein. As far as I know, there's not an eastern blot yet, but give us time. And another way of analyzing DNA is the microarray analysis. So with DNA probes, you have you've done your work with your DNA, you have your sample, you want to see if there's a gene, a particular gene there. You make a probe that has a complementary basis to the gene of interest. And then you mix that, you denature, so you get single-stranded DNA, so the strands separate, and then you add your probes to the same solution. And the idea is that the complementary basis here on your probe will bind to the area with the complementary basis of the DNA, which is in fact what is happening here. And what you do is you put some type of a label, oftentimes like a fluorescent label, where you will be able to see this. You do a rinsing process and whatever remains, okay, it's only if it's bound, so the excess ones that did not bind get washed away. And then you're able to say, okay, yes, my gene is here. Agarose gel electrophoresis. What you do here is you make your electrophoresis gel. You mix up your solution and then you pour it into the container and it solidifies. It has a consistency kind of like a Jell-O, a little bit firmer than Jell-O. When you pour the liquid portion in, you do put this is called a comb, this blue area up the top right here. And you stick that in. That's going to, as the solution solidifies, you're going to pull this comb out and it leaves these little wells in here. That's where you're going to put your samples into. So you put your samples into it and then you hook up an electrical current. And the idea behind this is, and previously your sample, usually you have added restriction enzymes to cut, say, the DNA up into fragments. So you want to see if the various fragments are there, which ones are present. With electrophoresis, when you hook up a current, the idea is that things are going to move. A lot of the fragments tend to be negatively charged, so they're going to move towards the positive charge. Opposite charges attract. Also, the smaller the fragment, the faster it's going to move. And so what you do is you also add, with each of the samples, you add a dye so that you can see it's a very low molecular weight dye. It's a very small fragment. So that's going to come down first. That's when you know when to turn the power off. So you turn the power on, the samples, they start moving down their lanes because you put the samples in the little receptacle holes that were made up here by placing the comb in. So the samples are moving down by charge, by size. Remember, the smallest ones go fastest. And then when it gets to the bottom, you turn the power off. And oftentimes what you do is you soak the gel usually in a stain, and then you look at it sometimes under UV light, depends on what stain you're using. And you notice a pattern. You will have, in one of the lanes, you always put some controls. And then you're going to look at the different fragment on here. You'll compare from one lane to another the different little fragments that are there. For the RFLP, once again, you're going to take some of the fragments, run through electrophoresis, and you're going to be comparing some of the bands here where there's differences. In the southern blot, like I said, you're looking at fragments. It's still electrophoresis. You look at fragments of the DNA. So over here, each one of these dark colored lines, that's the fragments of the DNA. So you can see if you've been doing some manipulations, did you get the fragment of DNA in that you want? Is it there? Microarray, showing the steps here, how you have two. In this case, you have healthy cells, cancerous cells. You go through the process, you add fluorescent dye so that the cancerous genes will glow red and the non-cancerous will glow green. So you can have several different samples here and look and do the comparison just by looking at the color. So how do you look at the proteins and analyze them from a molecular level? And this is where you're going to use what we know or commonly referred to as a page gel, polyacrylamide gel electrophoresis. This is what is the Western blot now. So very similar. You make your gel. You always start off with it in a liquid solution. You pour it, let it solidify. You have the comb. On this one, it's done upright versus in a flat tray. It just depends on which one you're using. You have your proteins that you have selected. You put your sample, load your samples on, hook it up to an electrical current. Once again, the larger fragments are going to be slower. Smaller fragments are faster. But it's the charge, because of the different sizes, it's going to separate those fragments out, and then you can compare from one sample to another. Polymerase chain reaction, PCR. This is a method. You do not have to know the steps of it. Just know that the purpose of it is to amplify your DNAs, to get an increase in the amount of DNA that you have. There are some modifications that have been done to this in the last several years. There's reverse transcriptase PCR, there's real-time PCR. Just to give you an idea with PCR, and I'm going to show my age here, is that when I was in school, that was before PCR had been developed. So I'm pre-PCR. So friends of mine that were working on projects that were genetics-based, where they had to have the DNA, they often had a limited supply of DNA. So they had to prioritize what experiments could they do because they only had a limited amount of DNA. That is not a problem anymore. You want to run a lot of different experiments, but you only have a certain amount of DNA. You take the DNA, you run it through PCR, and it's going to increase the amount or amplify the amount of DNA that you have, and then you can run all the tests you want. So it's been a huge, huge breakthrough for genetics research. Like I say, you don't have to worry about the process. Just know that what it is going to do is increase the number of copies. You go through what they call different cycles through, and so at the end you end up with a lot of copies of the DNA. Like I said, now you can do whatever test you want with it. So what can you do with this? Sometimes you get samples of DNA, and you want to do some comparisons. Okay, what do you mean? Well, maybe you have a patient who's gotten sick. In this particular example, you have a patient who has C. diff, Clostridium difficile. And you're trying to figure out, okay, there's different strains of this. And if you have, say, an outbreak, you're trying to figure out, who did he pick it up from? Which strain does he have? So you can take your samples, run it on a gel. And all of these gels, to get the fragments, you have to use the restriction enzyme to cut it up to get the fragments. Now, if you look at here, the patient, Javier, is on the bottom. And you compare with all these different strains that they have. And notice that the strain 027 is exactly the same as Javier's. That's the strain he has. The study of the entire genome is referred to as genomics. And there are people who specialize in this, where they may focus on a particular organism. Sometimes what they're doing is comparing the genome of one organism to another to see how closely related are they. When you analyze the genome, sometimes what you may be looking for is, okay, this is the genome of this bacteria. How can we develop an antibiotic that would be effective against this particular bacterium? Well, if we know what the genome is, maybe we can use that. We oftentimes are using diagnostic tools, making vaccines, other medical treatments, and we're also using it for cleanup of contaminated environmental sites. When you are studying the entire collection of the mRNA, that's referred to as transcriptomics, you're going to be analyzing the gene expression between infected versus non-infected cells. What's the difference between them? How does a cell respond to an infectious disease? If you know how the cell's going to respond, that may help you with treatment options. Monitoring the gene expression of various virulence factors. When does it get turned on? When does it get turned off? All that can help you with controlling diseases, identifying diseases, treating diseases. Sometimes what they will use for gene expression, in other words, to see whether a gene is turned on, is that they will add a certain protein that makes it very easy to see. Because sometimes it's hard to see how do you know the gene's turned on or not? Well, it's kind of hard to see sometimes. So a very common thing that they will use is a green fluorescent protein, referred to as GFP, that they will add. And in these pictures, it just shows some examples of it. In the far left in A with the mice, you can see that the two mice on the outer edges have some, under UV light, they have some green there. The middle one does not. So that means the gene that they are looking for, it is being expressed in both the outer two mice, but not the middle one. In the middle picture, it's a picture of some bacteria growing on a petri dish. And here you can see that, once again, under UV light, the bacteria are growing green because they are expressing, once again, that particular gene. Now, sometimes what they will do is they will add a different gene, and instead of glowing green, it turns it blue. So on this last petri dish on the far right, you can see that some of the bacteria are blue. That means they have the gene, they are expressing the gene. When I say it's expressing a gene, it means that gene is turned on, it's been activated. And they look blue in this case. So what are some of the applications of doing all of this recombinant DNA technology? How can we use this? We can use it to make new pharmaceutical products, maybe making new drugs, making new antibiotics. We can use it to make safer vaccines, subunit vaccines. Subunit vaccines are where you're not making a vaccine. You're not using the entire virus or the entire bacteria. You're just using a portion of it. An example is with the COVID virus. They made it just against the spikes on the virus, so they don't have the whole virus. That way, you're reducing the risk of you getting sick and getting the actual disease. You can't get the actual disease if you don't have the whole organism. So it's much safer that way. Some other examples of that they have used this successfully is in the production of making factor VIII. This is necessary for people who have hemophilia, which is a bleeding disorder. If they get an injury and they start to bleed, they could bleed out. So they have to receive an injection of factor VIII. Human insulin for diabetes, human growth hormone, and also the hepatitis B vaccine. Gene therapy is where we are looking at using clinical application of these genetic engineering techniques for curing diseases. Where an individual has an abnormal gene, the idea is to introduce a functional normal gene to replace that abnormal one. There are questions with a lot of this. There's pros and cons of everything. There are ethical questions just from the basics of, should we be doing this? If somebody has an abnormal gene, do we have the right to go in and put in what we consider to be the normal gene? That's a question, that comes up for debate. There are risks involved with this. If you add the functional gene in, anytime you add anything into the body, you have the risk of possibly triggering the inflammatory response. If the inflammatory response is mild, okay, but if it's severe, it could lead to death of the patient. Obviously, that's not what you want. You could infect healthy cells. There's concern of, especially if you're infecting healthy cells, could this revert and ultimately cause cancer? Oftentimes you're using a virus as the vector to introduce the new gene in. Is there a chance that that virus could become infectious and itself start causing diseases? Fine, you put a functional gene to replace an abnormal one, but now the vector made you sick with another disease. Does it inactivate another gene and now cause another problem because you inactivated a gene? So these are all risks and concerns that have to be considered. This is showing how they would do. This is taking specifically an adenovirus. Some of these cause cold, like common cold, etc. That you would put as you can see here, the DNA, you would put in the gene. You can see in here is the new gene that they've added in that's functional to replace the abnormal one. So you put that in the virus, you introduce the virus into the cell, and then ultimately this DNA is going to make itself, as you can see here, into the cell. Here's the new gene, so now hopefully you're getting the proper functioning of whatever the situation is. But as I said, there's concerns. There's pros and cons. That would be wonderful if you can do this to help cure someone who's suffering from some disease due to an abnormal gene. That would be wonderful. That's definitely a plus side. But is it worth it? You have to look at the risk. And sometimes, well, actually a lot of times in medicine, you have to look at things as we learn all these new technological advances that help advance the knowledge of science. They go hand in hand. But sometimes you also have to step back and say, just because I know how to do something, does that mean I can do it? And that's something where oftentimes then it's going to bring ethics and moral issues, and that sometimes is going to involve community involvement. Sometimes it's a personal decision, but it's something to keep in mind. Just because you can do something, does that mean you should do it? And that's something that each individual ultimately is going to have to decide for themselves.